Guys, today our lecture is about culture media. So we will discuss about this, that what is culture and what is media. First we will differentiate them and do their definitions. Culture media. Media. What is media? The food. The food material on which organism is growing is known as media. Is called media. So what is media? It is the food material on which organisms will be growing is known as media. And what is culture? Growing of the organism on food material. It is growing of organisms is known as culture of that organisms. Now for the culture media, we need requirements. So these requirements will be two. First requirement will be the food source. Two requirements will be food source plus matrix, because matrix is responsible for the formation of solid media. And second one is inoculum, bacterial inoculum. What is bacterial inoculum? It is the load of bacteria. Which is going to be used in the different culture media. Mean load of this is known as load of bacteria or bacterial inoculum. Now culture media are of two types. Types of culture media. Culture media. So there are two types of culture media. Either the culture with media will be liquid culture media, and either it will be solid culture media. Either there will be solid culture so two types of culture media, basically they are of two types, liquid culture media and solid culture media, and the third one is semi solid culture media, which will be in a jelly form. On the liquid culture media, there will be presence of only food source. Now requirements of the culture media, they are of two types of requirement. On the base of food plus matrix, culture media is divided into two, liquid and solid culture media. Now liquid culture media, here will be presence of only food source, while for making solid culture media we need both food source plus there must be presence of matrix, and this matrix will be agar powder. Agar powder, and this agar powder will be obtained from the seaweed. It is obtaining from seaweeds, which are responsible to make a media solid, mean it is responsible to make a liquefied media into solid media. Clear? Now components which will be required, the food components which will be required for the culture media. Now components which are going to be required for the culture media. Required components for the components of the culture media. Food components. What will be the food components? There must be presence of water molecule, there must be presence of food sources, mean nutrients, there must be presence of carbon sources, source in the form of glucose, dextrose, etc. Clear? Now there must be presence of sodium chloride, NaCl also, which will be as inorganic salt. Sodium chloride. There must be also presence of the peptones, and also there will be presence of the nitrogenous source, nitrogenous source, which will be in the form of beef extract, clear, which will be in the form of beef extract. Beef extract will be, it will be meat in the powder form meat. Powder, clear it. Now how to prepare, how to make, now we are going to discuss that how to make a solid media and how to make liquid media. Liquid media and solid media. For the preparation of liquid media, we needs nutrients, next water, inorganic salt, salts, but for making liquid media we don't need of any agar, because we don't want to make a solid media. Now what are the uses needs for the solid media? Liquid media is make within the test tube, and the size of this test tube will be broad as compared to the common test tube. And in this test tube all the nutrients will be present with inorganic salts and water. Now solid media, the solid media is, for the formation of solid media we needs nutrients as a food sources. Water must be present here, because for the culturing of bacteria we need water. Water is an essential component of the for the bacteria for life. Inorganic salts, also here must be presence of agar. Agar are of two types. One is known as Japanese agar, and it is of 2%. Its concentration is 2%, while the other one is known as New Zealand agar agar which concentrated 1%, concentration is 1%. And for making solid media we need petri dish. Petri dish. Why we are using petri dish? Because for making solid media we need broad area, broad surface area, and this is provided by petri dish. Clear? Now we are going to discuss about the culture media. How we are going now to make a media, either solid either liquid. So here we must have bacterial inoculum. And this bacterium inoculum will be taken by, and this bacterial inoculum will be taken by inoculating tube. Now how you are going to make liquid media? All the contents is present within the test tube. Now we are going to make a culture media, liquid culture media of a specific bacteria. We will take the inoculating loop first. We must sterilize. Inoculating loop consists of two parts. This one part is metallic part, and this one will be either wooden, either made by plastic. Clear? Now when you are going to make liquid media, you must take the inoculating loop, and the metallic part of the inoculating loop will be must be making sterilized by direct flame, and then will be dipped within the bacterial culture and will be dipped in the bacterial culture and will be placed and dipped within the liquid media, and then the liquid media will be must be covered tightly by the help of non-absorbent cotton. What will be the characteristic of this cotton or plug? It should be non-absorbent. Why it should be non-absorbent? Because if the cotton is absorbent cotton, so it will absorb the watery component of the liquid media, which will be not a good condition for the culture of bacteria. okay, because water needed by bacteria also for their growth. Done. Now for making solid media we are using Petri dish, and solid media are of three types. Either three type of solid media will be present. First one is called strict plate method. Strict plate method. Second one is called spread plate method. Plate, and the third one is known as pour plate method. Now each and every step will be discussed now. It's clear? Now we are going to discuss about the types of solid media, which are of three types. Yes? Media must be solid here, and the first one is strict plate method. Second one is known as spread plate method, and the third one is known as pour plate method. Clear? In all, not all of them, among all of these three, strict plate method and spread plate method, in the Petri dish there must be a prepare solid media, and all these media will be formed within the Petri dish. Now in this method, strict plate method, this is the bacterial culture, which is also known as inoculum. Inoculum will be taken by the help of inoculating loop. Now the inoculating loop will be dipped within the inoculum and will be bring in the strict method. First it will be putted in such zigzag pattern, in a zigzag pattern. Clear? And should be aware that the lines which you are going to be drawn will be at a distance from each other. Zigzag pattern. striking will be done by the inoculating loop pattern, and then it will be covered and placed in the incubator. And the time duration will be given for 24 hours, and the temperature will be also fixed, because temperature will be playing role here. Temperature will be 37 degrees centigrade, and then when you are going to take it and examine it, here will be presence of the cultivation will be present on those line which will be sketched by the inoculating loop. Why it is known as strike plate method? Because striking is done by the help of inoculating loop in this method. Clear? Now, spread plate method. In this method, by the help of inoculating loop, some drops will be put on the petri dish, and then by the help of spreader, which will be of such type, which is made up of glass. By the help of that spreader, you will spread equally all these drops throughout the petri dish on which the culture media will be present, solid culture media will be present, and be ensured that all the drops, all the bacteria which will be taken from the inoculum will be spread deeply within the solid media by spreader, and will be covered and placed within the incubator. For 24 hours, and time duration will be fixed. Time duration will be given 24 hours. It depends on the types of bacteria which you are going to be cultivated. Sometime it's 24 hours, sometime 48 hours. It depends on the type of bacteria. Okay. Now, temperature will be also given fixed, and the temperature will be 37 degrees centigrade. Clear? And after 24 or 48 hours, when you are going to take out it and examine it, it will give us lawn structure, which is also known as lawn of bacteria. Clear? And in the pour plate method, in this method, the nutrients plus inoculum will be present in a test tube. Both will be present in test tube. Suppose this is test tube and this one is also a test tube. Here is inoculum, mean bacteria, culture bacteria is present, and this is culture media, in which nutrients plus agar will be present, and you should be ensured that the agar will be dissolved by heat. Okay. Now, you are going to be pour Pour plate method mean at once, at a time, both will be poured, but ensure that the temperature of nutrient and agar. This solution must be not so heated. Why? Because due to excessive heat, it can destroy the structure of bacteria. They can kill the bacteria, and it depends on your practice, and you will feel them in your hand. Okay. Then when you are going to pour them, mix both, and then it must be covered and placed within the incubator for 24 hours, and the temperature will be also fixed, which is 37 degrees centigrade. And after 37 degrees centigrade, after 24 hours, when you are going to examine them, so it will give us different shades, that the microbes will be okay. It's understand now. The importance in all of them is given to the pour plate method. Why? Because when you are going to examine this culture media, so the microorganisms will be cultivated within different layer. Suppose we are taking a section from here. Look here. Some microbes will be cultivated at the bottom layer. Some microbes will be cultivated in the medial layer, while some of the microbes will be cultivated at the top layer. This is top layer, and this one is middle layer. And this one is the inner layer, bottom layer. Now, what you will get conclusion, that which type of bacteria are growing at the topmost layer, upper topmost layer. Those bacteria will be cultivated at the upper topmost layer, which are strictly aerobic bacteria. Mean for the cultivation, for the culturing of these bacteria, oxygen will be needed in lot amount. Strictly aerobic bacteria will be cultivated on the topmost layer, while in the medial layer facultative aerobic bacteria will be cultivated in the middle layer. Will be cultivated within the middle layer. Clear? And what about bottom layer? Which type of bacteria will be cultivated here? Here will be cultivated microaerophilic bacteria. What are the microaerophilic bacteria? These are those bacteria which need oxygen in very less amount. be sure that in this at this method of cultivation there will be no cultivation of anaerobic bacteria, because anaerobic bacteria, for the anaerobic bacteria oxygen will be just like a chemical and they will be destroyed, their structure will be destroyed within the presence of oxygen. Okay. So, on this method here will be no anaerobic bacteria cultivation, no cultivation of anaerobic bacteria. It should be keep in your mind. Okay. Thanks.